So let me jump right to my commercial. I figured I'd start off with the commercial break and we go from there because this is a real good example, I think, of the importance of science. And in this case, of course, the answers come from science, and of course, our government likes to say elsewise. And so really what we come down to is pay me now or pay me later. And I think this really applies to the climate change because I think this is a much more important problem than the virus. The virus is just sort of what I like to think of it as a preamble to the big event, which would be climate change if we keep going like we are. And basically, we always hear the argument, well, wait a minute, this is going to affect our economy if we try to adjust. And as we've seen with this virus, if we'd adjusted back in March, this would not have been a big problem, but we didn't. So as I say on the slide, the tiny, tiny virus brought the world's economy to its knees. Even more important to some people, I suppose, is the fact that football, the Big Ten has canceled football because 15 players have myocarditis. This is an inflammation of the heart, takes months to get rid of. These are young, healthy men, and they're going to pay a price. And the cardiologist that's with our tech team, the rumor I heard was that he is already withdrawn. He doesn't even want to be associated with this because this is a bad problem. Anyway, why two names? You see COVID-19, but I'll be talking about SARS because SARS-1 was the original name given to the Chinese virus 20 years ago. And really, we're talking about SARS-2. These are almost identical viruses. And you'll now hear about SARS-2D, which is the strain that came over to the West Coast of the United States, and the SARS-2G, which is a strain that actually happened in Italy. The mutation happened in Italy. So there's a change in the genetics of the virus. And already we have a new strain, and it came in through New York, and it's a little more infectious. So let me digress for just one moment because in this talk, I'm going to talk about DNA, I'm going to talk about RNA, I'm going to talk about proteins. So just a quick review. DNA is, of course, the code. Up here at the top, you see the DNA structure. DNA is found in all your nuclei and all your cells, and that codes for a total person. So if you took one cell, the DNA from that one cell is totally you. But how do you read this code? Well, it takes a protein to read that code. And what you see right here, this green blob, that green blob is a protein. It can move along the DNA, and it can make a copy in this new form, which we call RNA. It's slightly different, but not worth talking about today. So this is called a messenger RNA. That's what the little M stands for. The messenger RNA now is then read by a ribosome. The ribosome is also another protein. It's a protein complex. It sets down on the messenger RNA, reads that, but now it makes a protein. So what we have, the first is called transcription, and the second is called translation. We're translating the code of the RNA into the protein code, which is amino acids. So you can see this long chain of amino acids, and they fold up into a protein that then can do many different things. You have proteins in your muscles, you have proteins in your hemoglobin is a protein that carries oxygen around in your bloodstream. You have proteins that make hormones in your body. Some hormones are proteins. Proteins do all the work in your body. DNA just sits there and makes proteins. It makes the RNA. You can read that to make the RNA, and then you make the proteins. But the proteins do all the work. And what we're going to talk about in the virus is virus can't do this. And so here's a picture of the coronavirus. That's why it's called the coronavirus, because of this. You see the proteins on the surface. These are proteins out here around the surface, this halo effect here. And this is the young woman who actually discovered the coronavirus and gave it its name in 1964. She was, when she was 16, she was in Scotland. Her family was poor and she couldn't go on to college, so she got a job as in a medical lab. and she learned to really work well on the microscopes, and eventually taught herself to use an electron microscope, which is what you see right here. And she saw the original coronavirus and named it. Her name was June Almeida, and that was in 1964. And she named it because of that corona around it. She was thinking the corona around the sun. Now here's a whole bunch of viruses, different kinds of viruses. And you'll see that it says it's an infectious obligate. That means it's required to be a parasite. It has to infect the cell. The virus itself is very simple. It's just its outer shell, and then you'll either have DNA or RNA inside. So it's a DNA or RNA, and it's surrounded by a protein coat or a membrane coat. This virus up here, that's like a herpes virus. You can see it has a protein coat. These have membrane coats around the outside here. This virus doesn't have anything. Now, just to give you a little feeling for viruses, we live in a world of viruses. Now, here's one type of virus. This is a bacterial virus. Now, just to take that one virus, just that one virus, in the ocean, in the waters of the world, there are 10 to the 30th bacteriophiles. That's what they're called, bacterial viruses. That number is hard to imagine. It's 1 with 30 zeros behind it. That's how many of this one type of virus is in our water. So if you took a teaspoon of seawater, you'd have 2 million viruses in that teaspoon. Now, again, these are tiny. You can't even see with a microscope. And their mass is a femtogram. That's, now we do a decimal point, put 14 zeros and a 1. That's how much they weigh. And so it's tiny, tiny amount. However, if you take that 10 to the 30th, multiply it times that 10 to the minus 15 for the weight. So now you're determining the weight of all that particular virus. That is 1,000 times more than all the elephants on the earth. But this number here, I've always found even more impressive, that if you link this little tiny virus, you can't even see with a microscope, a regular microscope, head to tail, they will stretch 100 million light years out into the universe. So that sort of gives you a feeling that and the ocean is really a source of viruses. The paleo people studying viruses show that there's a common ancestor of these RNA viruses. Now, coronavirus is an RNA virus. Its ancestor is out in the ocean. And these viruses diverged a long time ago. Somehow, a spray must have been inhaled by a species on land, and then they became, they were able to infect species on land and started their life on land. Are you infected? Anyone in the audience? Well, you don't have to answer, but in fact, you are. We're all infected. The whole world is infected with viruses. What's interesting is you have your own virome. There on your skin, you have greater than 13 different viruses. In your digestive tract, you have greater than 19 viruses. In your bloodstream, greater than 19. Even in your brain, your nervous system, you have greater than 3 different kinds of viruses. They're all infecting your body. You're living with these viruses. We live in a cloud of viruses. We breathe viruses. We eat viruses. Viruses infect everything. Plants, insects, birds, fish, everything. Everything is infected with viruses. Viruses are part of our life, an important part of our life. In fact, viral DNA, the DNA from viruses, is part of our DNA. And it actually has a function in our body. So you're born with viruses from your mother, and they're detected as you. They're part of you. And they're in our DNA. In fact, as it says here, one viral DNA in our chromosomes allows the placenta cells to fuse, which is a crucial step. The placental wall is constructed totally of viral proteins. And they think, the information seems to indicate that the reason our brains were allowed to grow as large as they are is because of viral genes. They're an important part of our whole body. Now, a virus. Here's a virus, a picture of a virus. This is a DNA-containing virus. It has a protein coat. I like to think of them as dead. It's just a coat with the DNA inside for this particular virus. The virus, coronavirus, is an RNA virus. The virus can't do anything until it happens to bind to a receptor on a cell, and then it infects the cell. And then, this is the magical part. The DNA of the virus is read by the cell, and it makes the proteins for the virus, and then it takes over the cell. So is this the living form of a virus, and this the dead form? Who knows? But this cell is now a viral cell, and it produces viruses. You see the viruses coming out down here. Viruses weren't discovered until 1939 with the electron microscope. They thought viruses were liquids. Most people thought they were liquids. Pasteur always thought they were tiny bacteria, but this picture is the first picture of a virus. This is a bacterial virus. It's infecting a bacteria. This is a bacteria right here. And these are the viral particles infecting that bacteria. Now here's the important thing though. It's a parasite. As it says right here, it's a parasite. It cannot function until they replicate in a cell. And to do that, they need what's called the ribosome. Remember I told you the ribosome binds to the messenger RNA and reads out and makes proteins. That's the key element. The virus doesn't have this. That's why it's a parasite. It needs to have the ribosome to make the proteins that then make more viruses. So let's talk about the coronavirus. Here is a diagram of the virus. And these yellow knobs sticking out here, that's the corona part. And this is important. This is an important protein. As you see, it's really three proteins sort of wound together. See those three little knobs? It's wound together. This is the spike called the spike protein. And we're going to talk more about that in a minute because this is a crucial part of how the virus infects you. Now, below that, you'll see here a membrane. This gray thing running around here, that's the membrane because this is a virus. This is a membrane-contained virus because this virus buds off of our cells and takes our membrane. And this is the membrane of a cell. All the cells in your body have a membrane like this. You can see it's a bilayer. Here's one part, and there's the other part. And it's made of fatty acids. Fatty acids, and this is the key, all membranes are made of fatty acids. And what else is made of fatty acids? Soap. In fact, I used to see my great-grandmother make soap on her wood-burning stove. She'd take lard and render it on the stove and make soap. And that's why soap can come right into this membrane and disrupt it and essentially inactivate the virus. Soap does the same thing to bacteria. Bacteria have membranes around the outside. So it can, that's why soap works so well. That's why you wash your hands with soap to get rid of this virus. So it's wrapped up with a membrane just like this because it buds off the cell and has the same membrane structure. It'll have different proteins in its membrane. But there are 7 human coronaviruses. You see over here, they all came from bats. All these human ones came from bats. So the bats came down, they infected an animal, and then an animal, somehow that virus got into man. Now, these alpha coronaviruses, these two over here, they only make they give you the common cold. They're common cold type viruses. Here's 2 more. They also give you the common cold. They're in the beta coronaviruses. However, these 3 down here, these are the bad actors. These are the ones that we have to worry about. Now, initially, the bat gave SARS-1 to the civet cat. The civet cat was then the spillover animal into humans. Oops, sorry. The MERS virus, which is, MERS stands for Mediterranean, because it comes through the dromedary camel, again, from a bat to a camel, and then from the camel into humans. Now, these are really very lethal viruses. You can see the SARS virus, 8,000 cases, 772 total deaths. That's something like 20%. MERS virus is 33%. It's still around. This one is gone, we think. This one is still around and still affecting, coming from camels into humans. Now, what's the deal between SARS-1, that's over here on this side, whoops, I keep hitting the wrong thing here. This is SARS-1, this is SARS-2, this is our virus right now, COVID-19. SARS-1, the reservoir was, it came from the civet cat. What the Chinese government did was they went out and slaughtered thousands and thousands of these animals to cull the animals back. But the other thing that's important about SARS-1, even though it's more lethal, the symptoms show up before, right away. There's no transmission in the first 26 hours. They're at 24 to 26. Therefore, they could stop this in the hospital. So in China, as soon as a person showed symptoms, they put them in the hospital, isolate them, and this barrier, this sort of barrier nursing, would stop the spread. Whereas right now with COVID, first of all, we don't know what animal this actually came from. We know it originated in a bat, but it came through an animal. And the other thing is, as you know, people can be asymptomatic and have the virus. That's why it can spread so well. That's why we have this rapid community spreading of this virus. Now here, this is a better diagram of the virus. You can see that membrane, this is that bilayer membrane around the outside. The red spaghetti in here, that's the RNA. The blue, that represents protein, a protein inside the virus that helps to wrap it up. But I told you about the spike protein. That's this orange thing on the outside. This is the important part. This is the one that's so important for us trying to develop vaccines and to actually control this virus. So here again, this diagram, over here on the left, you see the yellow is the membrane, the purple now is the spike protein, the RNA inside. Now, this is a diagram of that spike protein. So this is the spike protein right here, setting down on a receptor on our cell. This receptor is called ACE2. That's angiotensin converting enzyme 2. Now, some of you may have high blood pressure and are taking ACE inhibitors. ACE1 is what that inhibitor inhibits. ACE1 and 2 work together to control your blood pressure. So this is a protein that you have normally in your body. All of your blood vessels have this protein, and it functions to help control blood pressure. However, by chance, somehow, this virus happened to use that as its receptor. The MERS uses a different receptor, but SARS-1 and SARS-2 both use the same receptor. So this is the one that happened to match the spike protein. So spike protein recognizes that and binds to it. This is the spike protein right here, setting down on this protein. It's another protein on the surface of your cell. So this is your cell membrane here, this sort of tan looking line. The viral protein membrane is up here, the yellow. And so what happens now, and this is an interesting process, it's clipped. There's an enzyme that clips this right here, right above those little purple balls, and leaves that part of the spike protein behind on this, and then this part jumps down and binds to your membrane of your cell. And now you have the spike protein locked into your membrane. But then an interesting thing happens. This spike protein, the stem part here, this is the stem part, this folds down. So you can see it folding. And as it folds, that brings the membrane of the virus down onto the membrane of the cell and allows this fusion to take place. At that point, the RNA of the virus can get inside your cell. And that's the process that's going on when the virus particle binds to your cell through the ACE first. It's clipped, it bends, brings its membrane down, and fuses. Now, what we're doing in our lab over here at the Health Science Center is we're actually making a little peptide, a little piece of the ACE2. We're making this little piece right here. And that will then, of course, bind to the spike proteins of all the viruses. And then to that little peptide, we're attaching selenium. So the peptide will bind to the spike protein. The selenium then has the ability to catalyze the formation of superoxide. And the superoxide will help to break up the membrane of the virus and essentially inactivate it. Essentially destroys the ability of the virus to infect you. And so this peptide, this little thing here that we're making from this, will be used to block the virus and to destroy its outer membrane. And so what we will do, we hope, this is actually being sent off right now to the government testing lab in Utah, and hopefully we'll know within a couple of weeks. If it does work, we think we can just put this in an inhaler and you will inhale 20 micrograms. 20 micrograms, if you take a pencil and dot it on a piece of paper, that weighs a milligram. That is 1,000 micrograms. So 20 micrograms of that, it's just a tiny, tiny amount, is all you have to inhale to inactivate all the viruses in your body. And probably would have to do this maybe once, twice a day for 4 days, and we hope you can get rid of the virus. So now don't panic when you see this slide. Look over here first. Look right there. Those are the 7 different human coronaviruses. And what's interesting here, this is the sequence of the amino acids that make up the stem part of the spike protein. And what's important here? Just look at the yellow. Just step back, sort of squint, just fuzz out. These letters here are the individual amino acids. But what the yellow shows is how they're identical, the identical parts. Here's a region right here. It's identical in all 7 of these viruses. And so this is a region that we'd really want to make our material bind to, because we could bind to all 7 of these viruses and inactivate all 7. Or let's suppose our drug doesn't work. If you want to make a vaccine, this is the region you'd want, because this is not mutating. These viruses have been around for a long time and no mutations. That's because this part of the spike protein is crucial. The other part of the spike protein changed. In fact, MERS is different from SARS. So that part, the part out, this is just the part, the stem part. These are 2 regions, those 2 regions that bound to each other. The reason they don't mutate is because they want to bind to each other. Here's another region down here, this one, that's all identical to all those different viruses. So anyway, for a vaccine, what is a vaccine and what do you have to do to get a vaccine? This is the key part here. A vaccine requires a protein. So probably the vaccine, I'm sure all the vaccines are being made of spike protein. That's the key protein, because that's on the surface. So that's the protein that your antibodies are going to recognize. So you would take parts of that spike protein or somehow get that protein, which is called an antigen in immunology, to signal the body to make antibodies, because this is a foreign protein now. Remember, you have a lot of viruses in you, but those are part of you. They came from your mother, and you don't make antibodies to them. But you do make antibodies to this because this is a foreign protein. So this antigen, the antigen we're going to use again, is the spike protein. So here's an antibody. This structure down here that looks like a Y, those are 2 arms of an antibody. It's actually a large protein. Again, all these things are protein. The protein, as you can see, has this little pocket up here on these 2 arms. And that's where the antigen, or in this case, the spike protein, the part of the spike protein would bind to this, and that will activate this antigen, or this antibody. Now, antibodies are made by B cells. B cell, B for bone, they're made in the bone marrow, the bursa of the bone. These B cells they can have antibodies attached to them, or the antibody can be free. However, initially it's attached. So what happens? This little B cell is floating around, and there are hundreds of thousands of different B cells, each with a different antibody on them. And if there's an antibody that happens to recognize and bind to the spike protein, then the antibody becomes the B cell becomes a little antibody factory and starts making lots of antibody. Some of them become a factory and others become memory B cells. And we'll talk about that in a minute. But the antibody factory now is going to make all these antibodies. The antibodies will go out, bind to the foreign thing, and then this recruits white cells from your body, like a macrophage, to come and destroy the virus. And that's how the vaccine works. So you can see why it's very important that we make a vaccine that's going to be very specific. In this case, the target's going to be the spike protein. We're going to have to make, hopefully get this to work in our body to make these antibodies. However, what's the effectiveness of vaccines? Well, you know, the flu vaccine, some years is 25%, some years is 30, some years is 50. That means 50% of the people are not really protected. And sometimes it doesn't work as well in old people. So sometimes they add adjuvants to make these things more active. Adjuvant can be something like aluminum is used as an adjuvant just to sort of rev up your cells to get them really going. Right now, they're talking about using animal antibodies. And the one they talk about is the llama. Why are they talking about llamas? Because llamas make a totally different kind of antibody. Their antibodies are tiny. They're just like the ends of those arms. They're 1/10 the size of a regular antibody, which means we could probably produce those in a factory. We could make those llama antibodies and inject those into people to try to knock out the virus. Now, here are all the different techniques they're using right now. This particular one, Baylor is using down at Baylor University, Baylor Medical School, I should say. They're making the spike protein. So they're using that. Here, this Novavax is actually attaching spike protein to a little nanoparticle to use that like a little dummy virus. This company, Codagenics, is actually using the Jenner technique. Jenner, when he made the, used cowpox a long time ago to generate antibodies because the cowpox would not, wasn't lethal for humans. So that's approach. This approach, using an adenovirus, that's the approach the Russians are using. They announced they had their vaccine. That's also the approach Johnson & Johnson's using, for instance. But the Russians haven't gone ahead and done their phase 2 or phase 3 trials, which we don't know what's going to happen. Other companies are actually using DNA and RNA in what's called little liposomes to infect you with that. So you'll actually make, your cells will actually make that. There's, at the moment, 276 vaccine candidates. So hopefully we'll get one. Although AIDS virus has been with us for 40 years and we still don't have a vaccine for that. Here's what happens. This is antibody production days after you're infected. They showed it increases, but then it starts to decrease. And in about 3 months, it's down quite a bit. And they're worried that you might be able to be reinfected. However, what we think is, remember I told you those memory B cells? Those memory B cells are sitting around there, and they could, if they're activated again, they can start making more antibodies. They think that's what's going to happen out here. So you will be protected. However, now we have this other thing going on, this American anti-vaccine movement. They say vaccines are rushed. Vaccine safety is not tested for properly, like with the Russians. Pharma and the United States government are in conflicts. And as you see over here, we have Operation Warp Speed by our government. So our government is moving too fast, probably, with these vaccines. They're pushing this fast. And of course, these people think that vaccines cause autism and other chronic diseases, which is not true. That's been shown not to be true. But right here in Texas, we have the health freedom anti-science. These people say they protest against vaccines, social distancing, contact tracing, even masks, which is ridiculous. So even though the virus is very tiny, those masks can stop it because the virus is in a little droplet, a little aqueous droplet, water droplet. But here's what's happening in Texas. 2003, 2,300 people refused to use vaccine exemptions for their kids to go to school. Now there's 64,000 people doing this in 2018. It's ridiculous. Here's one of our state legislators, Jonathan Strickland, saying things like this. He's saying this to Peter Hotz. Hotz is the guy at Baylor Medical School that's making the new vaccine. He's saying, parental rights mean more to us than your self-enriching science. So you see these people, this is a problem. So here we are, this epic struggle going on, vaccines and therapeutics against 20th century forces. One of these is climate change, and I'll mention that again in just a minute. Poverty, war, political collapse, urbanization, and anti-science. Anti-science is the biggest problem in this country. So what's the lesson so far? Viruses can kill anyone, young, healthy adults, even children. So children are not really that safe. They can have problems, long-term problems, from these little microclots that this virus causes. This virus does all kinds of things. Surfaces are not the big problem. It's airborne, these micro droplets, as I said a minute ago. So masks work. They're not perfect, but they do work pretty well. Many people are infectious without being sick, so that's a big problem. Vaccines are difficult to make, as we've talked about. Vaccines need to be made for people of different ages, but don't bet against science. So what's the deal here? This we usually talk about, you guys talk about climate change. How do these two threats converge? Well, the World Health Organization is predicting that climate change will give us a lot of new viruses due to the massive changes in the ecosystems. The European Society of Clinical Microbiology and Infectious Disease state that climate change is an ideal breeding ground conducive to virus modification. And that's the big problem. These viruses picking up mutants that allow them to become more infectious and cause more problems. This is a real serious problem. And so with that, I'll end. Thank you.